The animal is dead, but the food is already beginning to disappear. A gazelle has been dragged back toward camp. There is more meat than the group can eat today. The air is warm. Flies are arriving. Scavengers may be watching from beyond the edge of the firelight. If the carcass is handled too slowly, a large part of this successful hunt will be lost before anyone can use it. So the real survival problem begins after the kill. Without refrigeration, sealed containers, or modern preservatives, how did prehistoric humans keep meat from rotting? They did not have one universal technique, and they did not stop decay completely. They learned to slow loss, choose which parts were worth saving, and extend the time between an animal's death and a human meal. That changes what a successful hunt means. Finding or killing an animal does not automatically deliver all of its calories. A carcass is heavy, some tissues spoil quickly, some parts are difficult to carry, some are valuable enough to justify extra work. Others may have to be abandoned. A group facing a fresh carcass has only a few options. Eat immediately, carry selected parts away, process some tissues for later, or leave behind anything that costs more time and energy than it is worth. Every extra hour spent cutting and protecting the carcass also keeps people in one place, exposed to heat, insects, and competing scavengers. This is why archaeologists study which bones appear at a site. A nearly complete skeleton suggests one kind of behavior. A concentration of heads, limbs, or marrow-rich bones suggests another. The pattern can reveal decisions about transport, value, and timing. But the most obvious answer still seems to be fire. Cook the meat, and the problem is solved. Except, it is not. Cooking can destroy many microbes already present. It can change texture, taste, and digestibility. But cooked meat still contains a great deal of usable water. Once it cools, it can be contaminated again. In warm conditions, decay continues. Heating meat and preserving meat are related tasks, but they are not the same task. To slow spoilage for longer, something else has to change. The meat must stay cold. Salt or acidity must interfere with microbial growth. Or enough available water must be removed that many microbes can no longer multiply easily. That is why drying matters. Take one thick block of meat and one thin strip. Leave both in moving, dry air. The outside of the thick block may harden while the center remains wet. The thin strip exposes far more surface area compared with its volume, so moisture can escape faster. Wind, warmth, low humidity, and time can help. Rain, damp air, insects, contamination, and excessive thickness can make the process fail. Drying is not sterilization. It does not make meat safe forever. Some organisms can survive in low-moisture foods. But reducing available water can slow spoilage enough to create something fresh meat does not provide extra time. The mechanism is clear. The archaeological evidence is much harder. Meat, skin, wooden frames, cords, and plant fibers usually disappear. Tens of thousands of years later, researchers are often left with bones, stone tools, ash, stains in the soil, and small holes where posts once stood. They are trying to reconstruct a behavior after most of the behavior has vanished. Which body parts were brought into camp? Which were left elsewhere? Were bones broken immediately, or only after the outer tissue had dried? Do cut marks suggest fresh skin or dry skin was removed? Do bone concentrations match places where meat may have been processed? Do post holes belong to a drying structure, a shelter, or something unrelated? The first major clue in this story does not look like dried meat at all. It is a deer leg bone. At Kesem Cave in what is now Israel, people repeatedly processed animal carcasses between roughly 420,000 and 200,000 years ago. The site contains extensive evidence of butchery, selective transport, bone breaking, and fire use. Researchers examined deer metapodials, the long lower limb bones above the hoof. Some carried cut marks that differed from the marks expected when fresh skin is removed immediately after death. The marks were more consistent with cutting away skin that had already dried and tightened around the bone. That raised an unusual possibility. What if some of these bones had not been opened at once? A complete limb bone is not just waste surrounding marrow. The hard bone, wall, and the skin around it can act as natural protection. They cannot stop chemical change indefinitely, but they may shield the marrow from direct exposure better than a lump of muscle left in the open. Researchers tested that possibility with modern deer bones. They left metapodials exposed under controlled outdoor and indoor conditions, then examined how the skin changed, how the cutting marks changed, and how the marrow's nutritional quality declined. Some samples were tested after periods extending as long as 9 weeks. That number is easy to misuse. It does not prove that people at Kesem routinely stored food for 9 weeks. It does not mean exposed deer meat remained edible for 9 weeks. It does not reveal the actual storage time of any particular archaeological bone. 9 weeks was the outer limit of an experiment under specific conditions. The important result is narrower and more useful. The experiment showed that marrow inside an intact metapodial could remain nutritionally useful for delayed consumption, and that removing dried skin could leave marks resembling some of those found at Kesem. So one of the earliest strong cases for animal food storage may not involve making jerky. It may involve waiting before opening the bone. That is the first major reversal. Prehistoric preservation did not always require adding more technology. Sometimes it may have meant leaving a natural container intact. But marrow is only one part of an animal. It does not answer the larger question of what happened to muscle from a gazelle, deer, or other hunted animal. For that, the evidence moves much later, to a large hunter-gatherer site in eastern Jordan called Karaneh 4, occupied roughly 19,900 to 18,600 years ago. Here, researchers studied thousands of gazelle bones, their locations, butchery traces, hearths, and small post holes in the ground. 3 clues mattered most. First, particular gazelle body parts appeared in unusual concentrations. Second, small post holes could have supported light structures in parts of the site. Third, the bones, hearths, and activity areas formed spatial patterns consistent with organized carcass processing. Researchers compared these remains with models based on documented meat drying practices. Certain bone-in cuts are more practical to hang or dry than others. If people removed some meat, left other cuts attached to bone, and placed them on light structures, the discarded bones might form a recognizable pattern later. The Karaneh evidence is compatible with that idea, but compatible is not the same as proven. A post hole does not come with a label saying drying rack. It could have supported a shade structure, a work frame, or something else entirely. A concentration of ribs, vertebrae, or limb bones can reflect drying, but it can also reflect transport choices, ordinary butchery, dumping, or differences in which bones survived. Even a hearth nearby does not prove smoking. Fire could have been used for cooking, warmth, light, or tool production. This is the second major reversal. Archaeology may show that meat drying is a strong explanation, but it often cannot produce a single object that settles the argument. The behavior has to be reconstructed from several imperfect traces pointing in the same direction. That uncertainty does not make the evidence useless. It makes the conclusion narrower. At Karaneh IV, the bones, post holes, and activity patterns support the possibility that people were actively drying or otherwise processing gazelle meat for delayed use. They do not prove the exact shape of the structure, the exact method, or the length of storage. This is how deep prehistory often works. Evidence can make one explanation more persuasive than its competitors without making every detail certain. Later sites provide clearer evidence for other preservation methods, but they also create a new danger, assuming that later methods must have existed everywhere earlier. At Al-Qideh, in central Sudan, evidence from the 7th millennium BCE supports the use of salt to preserve fish. Researchers did not rely on fish bones alone. They examined mineral deposits and chemical traces connected with the archaeological material. At Norge Sunnensund in Sweden, dating to roughly 9,600 to 8,600 years ago, pits containing large quantities of fish remains, plant material, and evidence of seasonal capture support an interpretation of fermentation and storage. These sites show that prehistoric hunter-gatherers could build complex systems for preserving animal food. But they do not prove that people hundreds of thousands of years earlier salted gazelle meat or fermented deer. Fish behave differently from large mammal flesh. Salt availability varies by landscape. Cold northern conditions are not the same as dry eastern Jordan or Mediterranean Chasm. Their importance lies somewhere else. They reveal why the history of food preservation is so uneven. Salt crystals, fermentation pits, concentrated fish bones, and unusual chemical traces can survive clearly enough to be recognized. A wooden frame holding thin strips of meat may disappear almost completely. The archaeological record does not preserve every behavior equally. So when later evidence looks clearer, that does not automatically mean preservation suddenly appeared at that moment. It may mean that certain methods left better evidence. That is the third major change in the story. The history we can see is shaped partly by what prehistoric people did and partly by what time failed to erase. By this point, the search for one ancient preservation method has broken apart. There was no single prehistoric recipe. Instead, there were several control points in the race against decay. The first was selection. A group did not need to preserve an entire carcass. The most vulnerable tissues could be eaten first. Portable cuts could be carried away. Marrow-rich bones could be kept intact. Large, low-value, or difficult sections could be left behind. Preservation began with the decision not to treat every kilogram of the animal in the same way. The second was speed. Rapid butchery and transport reduced the time meat remained warm, exposed, and vulnerable to insects or scavengers. The third was water loss. In a dry environment, cutting meat into thinner pieces and exposing it to air could slow microbial growth by reducing available moisture. The fourth was natural packaging. An intact bone covered by skin could protect marrow until people were ready to extract it. The fifth was local opportunity. Where salt was available, it could become part of preservation. In cold environments, low temperatures could slow decay. In some later hunter-gatherer settings, fermentation turned seasonal abundance into food for later use. The sixth was accepting risk. Prehistoric preservation did not meet modern food safety standards. Delayed food could still spoil. Dry food could still contain dangerous organisms. A group was not making food permanent. It was trying to make loss slower than transport, sharing, and consumption. This is the mechanism that unites the evidence. Preservation worked when decay became slower than the group's ability to use the food. And that leads to the largest reveal. Without preservation, the death of an animal and the human consumption of that animal are trapped inside nearly the same short window. The carcass must be butchered, carried, shared, and eaten before heat, microbes, insects, or scavengers take too much of it. Delayed marrow extraction, drying, salting, cooling, or fermentation all do something deeper than change taste or texture. They partially separate the time when the animal dies from the time when humans have to eat it. That was the real breakthrough. It did not stop decay. It created delay. A successful hunt no longer had to exist only in the present. Some of its energy could be moved forward in time. At Kesem, the evidence suggests that intact limb bones may have allowed marrow to be consumed later rather than immediately. At Karane IV, the distribution of gazelle bones and traces of light structures are consistent with active drying or related processing, although the exact facilities remain uncertain. At later hunter-gatherer sites, salt and fermentation provide clearer examples of animal food being deliberately preserved for future use. The methods differed because the environments differed. The evidence differs because some behaviors left more durable traces than others. So how did prehistoric humans keep meat from rotting before refrigeration? They did not eliminate rotting, and they did not all use the same technique. They selected which parts were worth saving. They processed carcasses quickly. In suitable conditions, they could reduce moisture. In some places, They may have used light drying structures. They could leave marrow protected inside intact bones. And some later groups used locally available salt, cold, or fermentation to extend the life of animal food. What we can say with confidence is that at least some prehistoric communities learned to delay the consumption of animal nutrients. What we cannot say is when the first strip of jerky was made, whether smoking was universal, or exactly how long every method worked. Their achievement was not a perfect ancient refrigerator. It was learning how to make decay wait.